Drischer. I'm a professor at the Vrije Universiteit Brussels in Belgium, and it's with great pleasure that I'm inviting you today to join me for the Pfizer-sponsored symposium taking place at the World Federation of Hemophilia Virtual Summit 2020. Management of hemophilia in times of uncertainty, adapt, innovate, and evolve is our motto. So without further ado, I'd like to start off with some of the nuts and bolts, if you may, of gene therapy. How do we get genes into cells? It's easier said than done, but it took us already 3 decades to reach the stage where we are today. So the innovative approach for hemophilia care that I really like to highlight today is gene therapy. How do we conduct gene therapy? And these are my disclosures. I'd like to give an overview of recent successes in gene therapy clinical trials beyond hemophilia, and then highlight what kind of technologies do we need to actually deliver genes into cells. What are the dangers? What are the risks? What is the potential? And where do we stand with respect to gene therapy for hemophilia A and B in clinical trials so far? What can we learn from all this? What are the future considerations and challenges that we still need to address? Looking at the landscape of gene therapy clinical trials, it's really clear that the momentum in the field is increasing. There's long-term therapeutic effects demonstrated in a variety of diseases in many different studies affecting many different organs, the brain, the muscle, the liver, the blood system. All of these different disease targets have been successfully treated by gene therapy, resulting in the first FDA and EMA approved products. Not yet for hemophilia, and this will be the focus of my presentation. The increased momentum in the field hasn't escaped the attention of the policymakers, and when President Obama was still President of the United States, he was inspired by some of the successes in the field, also by other advances in personalized medicine, to launch what he coined as the Precision Medicine Initiative to bring us closer to curing diseases like cancer and diabetes. And Emily Whitehead, who is here on the picture, is a young girl who suffered from leukemia, who was treated successfully by gene therapy. To the extent that in order to launch the Precision Medicine Initiative, President Obama actually invited Emily to the White House for the inaugural event. In fact, she had a good excuse not to go to school that day. Coming back to hemophilia gene therapy, the current treatment for hemophilia requires factor VIII and factor IX substitution therapy. And there's a very safe track record with respect to these existing therapies. But it doesn't constitute a cure, and patients still are at risk of bleeds or joint damage or worse. But the bar is high for gene therapy of hemophilia, particularly because of these existing treatments. For many other diseases like Duchenne muscular dystrophy, spinal muscular atrophy, lysosomal storage diseases, hereditary blindness, just to name a few, there is no effective treatment. The goal of gene therapy versus standard of care is to achieve prolonged, if not lifelong, production of the clotting factors in the patients that are treated by gene therapy. If you look at the standard of care, patients are administered frequently with the purified clotting factors, factor VIII or factor IX, and the levels go up, but then reaches very low levels, requiring frequent readministration for the entire life of the patient. And this coincides, of course, with the differences in hemostatic efficacy. Sometimes the hemostasis is very robust, but eventually it also declines back to basal. With gene therapy, we'd like to achieve constant levels of these factors, hopefully physiologic, to the extent that we can achieve a long-term therapeutic effect. That is the ultimate goal of gene therapy. Now, the mantra of gene therapy is delivery, 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 to paraphrase the real estate mantra, location, 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 because it's all about getting the genes into the right target cells. And initially, the technology that we were using was rather primitive, as you can see here in this Larson cartoon. And we have improved on this technology over the past 30 years to the extent that we now have at our disposal fairly robust and also quite efficient and safe gene transfer technologies. So what are the technologies that we can use to deliver genes into cells? Well, we have to learn from nature here, and viruses that are very prevalent among us have developed very efficient means to deliver their own genetic cargo into cells. So we can harness that intrinsic ability of viruses to deliver therapeutic genes instead of viral genes into a patient's cells. So if you look at what a virus does, it infects a cell by interacting with the cell membrane and delivering the viral genes in the target cells, resulting in the production of viral proteins. These viral proteins will assemble to form an infectious viral particle that is capable of infecting new cells. Now, a viral vector harnesses that very potential of viruses to deliver genes into cells, but instead of delivering viral genes, the viral vector or gene delivery vehicle is adapted in such a way that the therapeutic gene, like factor 8 or factor 9, has been encapsulated in the viral particle, and it is that therapeutic gene that is then ending up in the target cell of interest. The consequence is that this therapeutic gene becomes activated in the target cells, the liver cells, for example, resulting in the expression of therapeutic factor 8 and factor 9 proteins. So there's no infection that is taking place here. There's no propagation of virus. We do harness the best of the virus to deliver the genes into the cells, but there is no inflammation or infection taking place. Now, what kind of technologies can we employ to deliver genes into cells? Well, we have actually two main viral vector technologies at our disposal, the adeno-associated viral vector technology, or AAV vector, and the lentiviral vector. And so far, all the hemophilia trials relied on AAV vectors, not on lenti, even though lenti also has significant potential, not just for hemophilia, but also for other diseases. AAV vector has the ability to give rise to long-term expression or production of the therapeutic proteins. Once the gene enters the cell, it doesn't really integrate in the chromosomes of the patients, only at very low frequency. So that means that these genes that are introduced in the target cells just hang around in the nucleus for a while, maybe even for several years or maybe even lifelong. I will come back to that later. Many individuals, however, have been exposed naturally to the wild type, naturally occurring AAV virus. That's very prevalent, resulting in more than 50% of the patients that are believed to be seropositive for AAV because they develop antibodies against the wild type virus that they have been exposed to. And this is something that we have to deal with, because if patients are to be enrolled in a clinical study or receive gene therapy products, they should not have antibodies to AAV. Otherwise, these antibodies will neutralize the vector. I will come back to that later. With respect to lentiviral vectors, these vectors have similar properties in the sense that they can give rise to long-term expression, but in this case, the vector integrates in the target cell chromosomes. So once you integrate, you're there forever. So the gene will never pop out, and this is a guarantee for lifelong expression of the gene of interest. No pre-existing antibodies to the vector components have been reported in most individuals with this type of vector. So this is sort of the yin and yang of gene transfer technology, if you may, if you're looking at AAV and lentiviral vectors. So with respect to hemophilia gene therapy, we're really aiming at delivering the genes to the liver. The liver is a natural factor VIII and factor IX producing organ in the body. And it's all a matter of finding the right molecular key to open the door, to deliver the therapeutic package into the liver cells. And relying on AAV as our gene delivery platform, there's many different variants of AAV we can choose from. There's a tremendous variety, diversity, a natural diversity of AAV viruses, each with a slightly different capsid that serves as the metaphorical molecular key to enter the cells. And with a different key, you can open different doors, and therefore different serotypes, you can enter different types of cells. So the distribution of the vector varies depending on these differences in capsids, and we call these serotypes. And these differences in AAV tropism is very important and may determine which serotype we choose to go into human clinical trials for which type of disease. However, differences in tropism doesn't necessarily translate to humans. If we see a tenfold difference in a mouse, it's not necessarily going to be the case in humans. So we do have to express some caution here at this stage. There's variation also in pre-existing anti-AAV antibodies towards each of these different serotypes, which is also something that we need to consider. And this variation in pre-existing antibodies depends on the nature of the serotype, but also on the age of the patients and the geography. So for in vivo gene therapy of hemophilia, we package the factor VIII and factor IX gene into the AAV vector. It's delivered through the circulation to the patients, and then in the end, the vector winds up in the liver where the factor VIII or factor IX gene is expressed in hepatocytes. Now, how can we maximize the efficacy of gene therapy? Well, we need to bet on 3 horses at the same time. We need to improve the vector, we need to improve the gene, and we need to improve the molecular engine, the promoter that is needed to drive the expression of the gene. So as you can see, this 3-pronged strategy allows us to improve the efficacy of gene delivery, improve the expression, and improve the functionality of the therapeutic gene all at the same time. And this is actually, this offers the best chance for therapeutic success. This increases the potency, allows us to lower the vector dose, and also minimizes the risk of side effects like acute toxicity and liver inflammation. Just to give you an example, the group of Paolo Sempioni and Walter Arruda and colleagues published a few years ago in the New England Journal the description of a mutation in the factor IX gene resulting in a gain of function, a super physiologic activity, if you may, which is now coined as the factor IX Padua transgene. And we and others have actually explored the use of this Padua gene in order to augment the efficacy. And it actually does augment the efficacy 5 to 10-fold, which is now confirmed also in some of the clinical studies. So what have we learned from all the clinical trials? Well, the lessons learned are that gene therapy for hemophilia can result in therapeutic factor VIII levels, therapeutic factor IX activity, a reduction in annual bleeding rates, a discontinuation of prophylaxis, and that these levels are relatively sustained for at least 1 year. No antibodies to factor VIII or factor IX were reported, but there are also some issues that we still need to address. There's patient-to-patient variation. Some patients have more than 100%, others have less. There's also issues of transient liver inflammation that need to be dealt with. The million-dollar question is, will expression be lifelong in patients after gene therapy? We know that in mice, hemophilic mice, the expression is lifelong. The mice that we have in our labs, they die of old age. They didn't die of hemophilia any longer after gene therapy. In dogs, it's at least 12 years. In young human primates, more than 6 years. The human experience is, of course, much more limited, but we now know that liver-directed gene therapy results in therapeutic levels of factor IX for at least 9 years. There are earlier muscle trials giving rise to expression for at least 10 years, and the clock is ticking. We do not know how long this expression will last. The issue here is that the hepatocytes, if they do not divide, and since this vector doesn't integrate, we expect to have lifelong expression in a non-dividing cell. However, if there is slow hepatocyte turnover, then since the vector is not integrated, there will be a dilution of the non-integrated vector genomes encoding factor 8 and factor 9, and this will result in a slow decline in expression. Since the liver can be subject to events that trigger liver cell proliferation, this may be an issue that we have to deal with in the future. Another important issue is the question of antibodies to AAV. Once a patient has been treated with AAV vectors, he cannot be retreated because he has then neutralizing anti-AAV antibodies that block the transfer of genes into the hepatocyte. The same thing happens following natural exposure of individuals to wild-type AAV. These naturally occurring antibodies block viral entry in hepatocytes. But perhaps there are ways to deal with this, and in the future, we may have to rely on immunoadsorption to get rid of these antibodies and then still enable AAV gene transfer to occur. And there's some studies in non-human primates that actually illustrate the potential of this approach. So in conclusion, we can say that gene therapy is effective. It's approved by regulatory agencies for some types of genetic diseases and cancer. In the context of hemophilia gene therapy, we know that it's lasting for at least 9 years. We do not know yet if it's going to be lifelong, but there are also other tools that we could consider, like integrating vectors or gene editing, that would give rise to a more permanent genetic modification of the liver cells. What are the main safety concerns? Transient liver inflammation, which requires corticosteroid treatments, possible long-term risks of random genomic integration. We do not always have a way to control where the gene integrates in the genome, and this may evoke certain risks. There are unknown unknowns and known unknowns to consider, and AAV seropositive patients cannot be retreated at the moment, but there's definitely certain approaches that are being developed preclinically that perhaps will ultimately find their way into the clinic, allowing seropositive patients to be treated. Can we ultimately treat children by gene therapy, ideally before the onset of joint disease? Well, for the moment, not yet. Again, we may need to rely on an integrating vector or gene editing, or perhaps develop strategies that allow vector readministration. The moral of the story is that this is not the end, to paraphrase Winston Churchill. It's not even the beginning of the end, but it is perhaps the end of the beginning. And I'm certainly more optimistic than ever that we will make a difference in patients' lives, not just for hemophilia, but for many other life-threatening diseases as well. Thank you very much. And it's my great pleasure to introduce the next speaker, Dr. Wolfgang Miesbach. Dr. Miesbach is currently the head of the Department of Coagulation Disorders and the Comprehensive Care Hemophilia Center at the Goethe University Hospital in Germany. He's a principal investigator for a number of ongoing clinical studies, including hemophilia gene therapy. I've had the pleasure to discuss and interact with Dr. Miesbach on many different occasions, and I always appreciate his nuanced perspective. I'm delighted to have him here today and welcome him to share his experience on facilitating the evolving conversations between patients and clinicians. Thank you very much, Dr. Miesbach. The floor is yours.
Thank you very much, Thierry, for this very kind introduction. I also would like to thank Pfizer for inviting me to give this talk in this very interesting symposium about some questions resulting from the basic scientific findings Thierry presented and now the discussion which is up between patient and doctors, and I would like to mention some key points which would be very important into this discussion. Now these are my disclosures, and now I would say we now live in an area of new hemophilia treatment. The new medications, the new treatment have the possibility to replace the current standard of care we have in the treatment of hemophilia. There are new ways of actions, new administration forms, and also the possibility to better individualize the treatment of hemophilia. And of course, gene therapy has the potential to treat hemophilia with one single intravenous administration. Now, the promise of innovative therapy in hemophilia is less burdensome and to offer more efficacious treatment. With this new options, the limitations of existing treatment can overcome, and it could be offered a durable bleed-free period with no spontaneous bleeds, no traumatic bleeds, and even no bleeds during surgery. With this, there would be no need for additional treatment with factor replacement, and most important, we would like to see no major adverse events with new treatment options. Now, if we have this discussion with patients, there would be several questions. And all of these questions are really not easy to answer. For example, the first question, how gene therapy works. Now, we all are no specialists for molecular biology. And but we can explain that gene therapy works like a gene transfer to replace the disease-causing gene by a functional copy. And this is a one and done treatment. It's not reversible, and it's more easy to explain how the gene transfer will be administered. This is the intravenous infusion, which takes from 30 minutes to 2 hours. What is most interesting for the patient would be the possible clinical benefits. So how it works, how long will it take, what will I benefit from? And I would say this is the most important question, a key question to discuss gene therapy with the patient and also to manage the expectation of the patient. What differs to other treatments we are used to is the frequent clinical follow-ups which are required, especially for the first weeks and months after gene therapy. There would be a variability of results. It's not very easy to predict. It's not impossible to predict the results. And of course, there could be some potential risks, short-term and long-term. And also vector shedding should be discussed. That means the transmission of the donated DNA fragments. So obviously, gene therapy will have a great impact on the daily life of the patient. But gene therapy would not be one solution for all patients. There are some limitations so far, so a lot of exclusion criteria, and even in patients who would be ideal candidates for gene therapy, maybe they do not want to undergo it. So there are many issues that healthcare practitioners must discuss with the patient, because gene therapy differs from any other treatment we have so far in hemophilia. There are many issues that patients must consider. They should have enough time to discuss these issues with their doctors and with their families. And they should know this is a unique challenge, a one-and-done treatment, which cannot be reversed. Now, according to the existing trials, there are issues of limited patient eligibility. So far, no children or adolescents have been included because of possible dilution effects, and also very old patients are not included so far. As well as patients with inhibitors against coagulation factor, and even patients with a history of inhibitors have not been included. It's quite clear gene therapy is a liver-targeted therapy, that severe underlying liver diseases are an exclusion criteria, but patients who had been treated successfully because of hepatitis C or hepatitis C, or even are under controlled HIV infection, may be treated with gene therapy. Another limitation so far are the presence of antibodies against the vector, against AAV, which makes it difficult to have an efficacious gene therapy treatment. But the gene therapy is evolving, and these inclusion and exclusion criteria may change in the future. There are even now some studies planned for patients who are adolescents, for patients with inhibitors, and there are already studies ongoing for patients with the presence of AAV antibodies. Now, most important for the patients, a possible clinical benefit, and this has been demonstrated in this study about patients who participated in a hemophilia B trial using the Padua variant. And as it can be seen here, nearly every item of the HemoQol quality of life questionnaire improved, mostly significant, even 1 year after gene therapy. Mainly sportive activities, physical health, even family planning and relationships. There was only one item which did not improve. It was even worse. This was the item dealing with the disease. And this may have something to do with the uncertainties and the unknowns of gene therapy. Even after successful gene therapy, we will come to that point later on. Now, for example, it's not possible to predict a successful gene expression in gene therapy. So gene therapy may vary, the result of gene therapy may vary from patient to patient, and there are several patient-related and also treatment-related factors which also vary and which may lead to these results. So especially treatment-related factors are very important. There are many manufacturing processes, many different capsids and gene cassettes, which also may have an impact to the immunological response of the patients. And it would be one very important thing in the future, that these different aspects will be discovered more closely to have the possibility to predict the success of gene therapy. Now, one very important point is the question of retreatment. So it may be that even after successful gene therapy, the gene expression of the factor would be less. And for this, there is one main limitation. This is the presence of antibodies against AB. So again, in gene therapy, we are faced with a problem regarding immunogenicity. So as Thierry already explained, AAV are an ideal vector to bring the gene into the cell. They are non-pathogenic and only weakly immunogenic. But the problem with these AAV vectors is that many of us have already been exposed to this vector and have an immunogen, so that these vectors do not work anymore. In any case, after gene therapy, patients would develop high titers of antibodies against AAV, which makes it impossible to re-administer gene therapy successfully. And it would be very interesting to develop in the future some options to overcome this limitation. One other problem we see in nearly any trial of gene therapy are side effects which develop very soon after start of the treatment, between week 5 and 10 mostly, but it can also develop later. This is a kind of T-cell reaction leading to ALT elevation. This is a liver marker and to transaminases. In all cases, this ALT elevation was transient and it could be treated successfully with short doses of steroids. But the problem of this T-cell reaction is that it can lead to reduced factor expression. That means that patients express very good factors, but after this ALT elevation, factor reduces nearly to the factors the patient had before. So the success of gene therapy could be in danger after this ALT elevation, and therefore it's very important either to prevent it by prednisolone or to treat it as soon as possible. Now, what we know about gene therapy has the potential to improve the quality of life, to prevent bleeds, but there are some points which are still unknown. And this makes it very important to discuss in front with the patient. So it would be very important to discuss with the patient which factor levels could be achieved. So, for example, which factor levels have been achieved in the past with similar gene therapy. How to overcome, if it's possible, the interpatient variability. ALT elevation is not unfrequent, but it can be treated very good with steroids. The next point would be the vector shedding, the durability of results, the long-term safety. Thierry already mentioned that now we are able to oversee a period of up to 10 years, so this is quite long, but what will be in 15, 20 or 30 years, we do not know, and maybe there are other unknowns we do not know at all. I think it's even much more important to advise the patient and to spend a lot of time to discuss things with patient after gene therapy. So patients are not used to a bleeding-free period without replacement of factors. And this can not only change the life of the patient, prevent bleeds, and lead to joint health and relief of pain, but it can also cause a potential anxiety and emotional issues resulting from these changes in hemophilia management they have not experienced before. So it would be very important to have a quite close contact with the patient after successful gene therapy. The patient should be aware how bleeding risk is likely to change, and any increase in physical activity should be taken very cautiously. It should also be discussed with the patient that there would be an option in a small proportion of patients that gene therapy would not lead to the results which are expected, and the patients still have to be on replacement treatment. Another point would be that any precautionary requirements should be discussed to avoid shedding of gene particles. Very important would be to advise not to use alcohol consumption, because this can damage the liver, and we also should prepare the patient for the need for quite frequent study visits the first time after gene therapy, and also emphasize that lifelong monitoring would be mandatory after gene therapy. So please let me summarize. With the progress of gene therapy, more and more patients are now treated successfully. We do know that gene therapy works in hemophilia. This is great. But shortly after gene therapy, there is the need for more regular follow-up. And there are some restrictions in daily life patients maybe are not used before. Later on, the lifestyle can change. The patient become more active, and it also could become difficult to convince patients to regularly visit the hemophilia treatment centers for regular follow-ups. We should engage patients very clearly and further follow up with some education to set the right expectations of the reality of gene therapy. And finally, COVID-19 has taught us the importance of the continued patient engagement even during this crisis, that we further on can recommend and follow up our patients very closely. So thank you very much for your attention. I would be very happy to take any questions.
Okay, our final speaker is Lawrence Woolard. Lawrence is founder and director of On the Pulse, an independent consultancy providing specialist insight and strategy expertise in patient education and engagement to global healthcare providers committed to hemophilia and other rare diseases. Lawrence's personal journey of living with hemophilia and its impact on the family dynamic motivates and inspires him to push for change in patient care. I'm really pleased to welcome Lawrence to share the patient perspective on evolving conversations between patients and clinicians. Thank you, Lawrence. The floor is yours.
Thank you very much for that introduction, Thierry. Hello and good afternoon, everyone from the UK. It's an absolute pleasure to participate in this virtual symposium. I would like to congratulate the sponsor Pfizer and their agency partner Synergy for organising this today in such a short space of time and for inviting me to take part. It's a personal honour to share a platform with both Thierry and Wolfgang, and to have the opportunity, as Thierry mentioned, to give the patient viewpoint around enabling evidence-based patient choice as part of the decision-making process in this current era of transformational therapy. So again, as Thierry mentioned, I manage my own consultancy, providing insight and strategy expertise in patient education engagement to the global pharmaceutical and healthcare industry. In the interests of transparency, this year I've engaged directly with these companies in a non-clinical capacity. So it's often said that music is food for the soul, and I, for one, have really been needing to nourish mine with an ever-growing playlist while on lockdown. Like many people, music plays a prominent role in my everyday life. It's deeply rooted in human nature and is a universal language that transcends cultural boundaries. It can also be a means of self-identification, especially in adolescence, where musical choices act as a badge of honor, satisfying social, emotional, and developmental needs. And this speaks to the photo of me, aged 11, taken in my grandmother's garden. Now, this was at the height of emo rock. I was a huge fan of Linkin Park, telling from the T-shirt, and also the dyed black hair that I somehow persuaded my mum to do for me. More than anything else, this was a form of self-expression, and I was using music as a type of coping mechanism, living with haemophilia, possibly reflecting the desire for acceptance with non-affected friends. And in fact, I would actually go on to study and work in the music industry throughout my early 20s. If you love music like I do, then you may appreciate that the way we consume music and engage with our favourite artists has rapidly changed due to technological innovations, including digitalisation, content streaming platforms, and social networks. And in today's music industry, there is more choice to both artist and consumer than ever before. Anecdotally, this enabling process is a reflection of what some patients and healthcare professionals have witnessed in the changes in management of haemophilia through advances in treatment and the revolution in digital health technologies. On screen is 6 decades of inspiration and innovation, using the music listening experience as a reference point for the dramatic rate of progress in haemophilia care. The arrival of cassettes in the early 60s, clearly way before my time, changed the entire landscape of popular music. In haemophilia, Judith Paul's fortunate discovery of cryoprecipitate is said to have paved the way for the modern management of care in the 70s. Home treatment was deemed a gigantic leap forward, as was Sony's Walkman that heralded the age of nomadic and private music listening on the move. Growing up in the 90s, I would gain access to safer recombinant treatment. In music, illegal file sharing was all the rave, literally. And from the early millennia and into the last decade, a new world of music would emerge, dominated by online streaming platforms and the explosion in user-generated content. Likewise, in this period, our community would witness a new frontier in haemophilia care with the arrival of EHLs, the first non-factor agent, and the real prospect of gene therapy. All in all, this timeline reflects a rich tapestry of stories, images, tragedies, and successes. So what's driven this innovation in haemophilia? Now, Wolfgang referred to it earlier, and as you may expect, much of it has been born out of the necessity to improve clinical outcomes and quality of life, as well as by the legacy of the contaminated blood tragedy. As detailed here, haemophilia can impose multiple complications, and there is an ongoing need for solutions to reduce the longer-term impact experienced by me and my peers. The pace and complexity of innovation to address some of these and overcome the limitations with conventional therapies, however, is also opening up new dilemmas with regards to upskilling people living with haemophilia and improving knowledge acquisition to support individuals and their families make treatment choices. Now, there is some qualitative and quantitative evidence in haemophilia of patient understanding, expectations and perceptions of newer treatments in countries with developed healthcare systems. In my opinion, the 3 key things that stood out from these particular papers on screen were, firstly, the need and request for better and more information to address identified patient knowledge gaps, such as understanding PK and optimal regimen on EHLs, the benefit-risk ratio before switching to a non-factor therapy, as well as awareness and strong safety evidence of gene therapy. Secondly, calls to support with patient-physician interactions, so people living with haemophilia are able to have a voice in decisions around new treatments. There were also natural concerns regarding the cost and reimbursement of current and future therapies. In summary, these bring attention to an overriding necessity for delivery and equity of access to high-quality patient education to enable them to make well-informed, meaningful choices and actively participate in their care. Now, based on my past experiences in music, it's interesting to note the similarity between how the relationship of the artist and record label have changed in recent decades, like that of the patient-physician. Today's musicians operate in a do-it-yourself digital world that offers them new opportunities to create their own path to success. The proliferation of online tools for recording, distribution and promotion means artists are enabled to produce music literally in their living rooms and reach a global audience by themselves. Radiohead were one of the first bands who tapped into this new opportunity. Digitization has restructured the power dynamics between the artist and record label. When once the label was the dominant gatekeeper to an artist's success, with the artist not having much say, the artist now has greater autonomy with a network of services and platforms at their disposal to control their own destiny. And you don't even have to be a big commercial band. An example within our community is my friend Liam McLean from Scotland, who lives with severe hemophilia B and an inhibitor, pictured bottom right of the screen. He's an awesome composer and multi-instrumentalist and has released his own material through Spotify. And I promised I'd give him a plug, so please check Liam out through his Instagram. So the patient-physician dynamic has been shifting in the same direction. Once traditionally based on paternalism and hierarchy, is moving to a more patient-centered approach. This has been facilitated not least by the growth of web-based health-related content and online transgeographic communities for people with the same condition, providing more access to information, thus giving rise to the empowered patient or e-patient. The diagram on screen is adapted from the Health Foundation's framework for patient-centered care. They suggest that whatever the specific care or interventions a person receives, it should be done with these key principles in mind, along with treating the person with dignity, compassion and respect. Importantly, they make the distinction that for care to be enabling, the relationship between healthcare professionals and patients needs to be a partnership rather than the patient simply following the professional's instructions. It's been suggested that a patient-centered care model is one where the physician's role is to be an expert advisor while the patient is an active, responsible co-worker. However, patients' drive to empowerment lies in their motivation, interest, health and digital literacy level, among others, in finding, conceptualizing and using the right information to improve their health. Also, it is easier for some people than others to assume more responsibility as part of the shared decision-making process. Hence, putting this into practice requires people to be offered support tailored to their needs, preferences, and level of activation. The concept of patient activation has been helpful in understanding why some people are active in managing their health and others are passive. According to Hibbard et al., patient activation underpins most health-related behaviours, including health information seeking. Research shows that higher activated individuals are more proactive about finding health information and using it in choice, as well as more likely to perceive benefits associated with shared decision-making, ultimately achieving better health outcomes and improvements in the overall care experience. And this includes high-quality interpersonal exchanges with healthcare professionals. In short, as Hibbard et al. Suggests, when people are active participants in their own care, use evidence to inform their choices, and when they understand and demand high-quality clinical care, they can be potent agents for change. Within the haemophilia clinical setting, new models of patient-centered practice are being realised to provide education, improve engagement, and encourage open patient-physician dialogue. In Canada, the patient is invited into a meeting with all members of the healthcare team to promote freedom of participation in treatment decisions, enhanced by the use of appropriate visual tools and decision aids. Gu et al. Report how their co-piloting approach supports the autonomy of patients through these 4 methods on the screen. Above all, as the below diagram illustrates, their partnership journey is a patient-driven process with patient priorities central to true informed shared decision-making, building a foundation towards successfully reducing or eliminating bleeds. Now, when it comes to information provision itself, whether in clinical or at a community level, not only is it necessary to take into account people's level of knowledge, skill and confidence for managing their health, but there is also generational differences and trends at play. The basic notion of generational theory is that we are shaped by a set of experiences, typically defined by a birth year range, which influences our collective thinking and leads to the acquisition of broad and common values and beliefs, primarily adopted during the formative years of each generation. It may seem obvious, and generational cutoff points aren't an exact science, but understanding unique attributes, characteristics, needs and preferences of these segments can help drive decisions about content and format when developing and delivering educational resources and programs. In particular, those born from the mid-90s have grown up in digital, with youth audiences accessing their media at the touch of a button and social networks having considerable significance in the lives of contemporary young people, including negotiating their identity and social status amongst peers, as well as the social criticism and political engagement of movements highly significant in young people's cultural lives. For example, climate change, gender and race equality. To this end, gaining deeper awareness of a different generation's frame of reference will serve to bridge the gap between young people in particular and healthcare professionals who are trying to build relationships with them. Of particular note, I think, is the societal impact of the coronavirus and the prospect of young people facing declining social mobility, which the think tank, the Resolution Foundation, has referred to as the Corona class of 2020. Before I bring my talk to a conclusion, whilst I've explained how it's easier than ever for musicians to get their music out, what's difficult is building awareness for one's music in a market flooded with new releases. To build a fan base, it's important to understand what cross-generational audiences and consumers want. Point being, an audience listens and then walks away, whereas a fan buys in for the long term. Using this as an analogy for engaging people living with haemophilia in and outside of the clinic, to mobilise an active network of superfans, or in our case, super advocates, we can't just put content out with no one in the room and hope for the best. Any information intervention first has to build in a consistent and compelling use value and consider where the value is best placed across these touch points that's most relevant and relatable. Simply put, for patients to derive value from content, there has to be a sense of purpose for why they should engage with it, continue returning to and investing their time in the platform or source, and be inspired to go beyond expectations by encouraging other members to do more, connect more, and engage more in the community in which they celebrate. So in closing, I've highlighted some key points of consideration to inform strategies for educational content and output around new treatments. In keeping with the music theme, like artists, success is all about reaching new audiences. And I think it will be important to experiment with alternative approaches and multi-channel content to support outreach and communication. When considering the audiences, creating unique, immersive and participatory experiences that challenge conventional ways of delivering education is necessary. And we're seeing examples of this already by using music and the arts as a vehicle for peer interactions to promote learning. It's my opinion that for the patient being asked, do you have any questions in respect of new therapies and their broader care, is disingenuous and potentially anxiety-inducing if they have not been exposed to appropriate learning opportunities to become engaged and activated to help make an informed response. Without investment in age, developmentally appropriate and culturally relevant education and effective delivery thereof, efforts to enable patients to participate in shared decision-making risk becoming a tokenistic gesture, reifying age-old power differentials with patients positioned as passive recipients of their care. On that note, I'd really like to thank you for listening.
I wanted to thank the speakers, Professor Misbach and Mr. Woolard, for their well-balanced and sometimes even thought-provoking presentations. I also wanted to take the opportunity to thank Pfizer for inviting the three of us and for also organizing this satellite symposium. And of course, the WFH for making all of this possible. We are now ready for the Q&A section of this interactive symposium. Maybe I can start off with some of the questions that came from the delegates, and I truly appreciate the delegates for participating in such an active way. We've had huge response rates. We cannot address all of the questions, obviously. It will take a lot of time, but I will select some of them here. One of the questions pertains to the future of gene therapy trials after COVID-19. And I would like to point a question to Professor Misbach. So what are the repercussions of COVID-19 that has impacted on all of our lives, but particularly now in terms of the future trials for hemophilia?
There was a very slow development the last few weeks and months. So in our case, so we stopped nearly every new patient coming to trials, and we also tried to have a very close contact to our patients, especially the severe patients. So with phone calls, we introduced video conferences, which was really a very great idea. This was very well appreciated by the patients. But regarding your question, no, we do not know how long this pandemic will go on. But nowadays, so especially when I can speak for our situation in Germany, we now have opened our ambulance for patients, and also I think trials can go on, but it's very important to fulfill all the requirements to prevent infections between the health professionals and patients, and it's a very difficult situation at that time, but I'm quite optimistic that at least as it seems today, for the situation in Germany at least, that trials can go
on. There's another couple of questions which are actually related to alcohol consumption. At one point, it was mentioned that alcohol should be avoided, and there's tremendous interest for how long this abstinence should be implemented. So do they have to avoid alcohol for their entire life? And I think this is again a question for the clinician on the panel. So Professor Misbach.
Yes, again, a very good question. So, use the liver cells for gene expression, and so it would be very important that the liver is in a very good mood. Sorry, I think there are some technical problems. Okay, so I think it would be very important not to damage the liver function. And the next question, which also would be very important, how long the gene therapy will take. And we are, I would say, a little bit surprised that we see expression rates so now for 10 years or even longer. And for this, it would be very important to protect the liver function and not to damage it. And I think, as we all know, alcohol, it's always a matter of the dosage as well. And I think everything should be done to protect the liver from damage, and this, of course, can be done with very, very careful use of alcohol. The first few weeks and months, and even the first year after gene therapy, I think no single drink would be allowed. This is my personal meaning.
Pertaining to the gaps and opportunities to support clinical colleagues with educating their patients around new therapies in the clinic. And this is in light of the ISTH International Survey that was also published by Peyvandi et al. It's also a very relevant question. I think maybe I take that one. There are always gaps in our understanding. This is a work in progress. And as a basic scientist, we are trying to unravel what these gaps really are, the unknown unknowns, and shed light onto improving the efficacy and safety of gene therapy. Socrates, the famous Greek philosopher, once said, the more we know, the more we realize we do not know anything. And I think it's the same motto in life and research, and certainly also gene therapy. Education is key. I think it's important to involve all the stakeholders as we did today. We have patients, we have the entire patient community, physicians, basic scientists, the industry representatives. It's only by coming together and communicating that we can ultimately come to a better understanding to achieve our common goal towards a long-term, safe and efficacious treatment of hemophilia. So the next question is perhaps of a more personal nature. So with respect to Mr. Woolard, so what would you need to see in order to be convinced to enroll in a gene therapy clinical trial, taking into consideration that there are, of course, other products available right now?
I was worried about you asking this one, Thierry. But no, it's a really good question. And first of all, obviously, I've really enjoyed observing the symposium. And again, thank you for the invite. I think there's actually been some really great talks given during the summit so far, and particularly yesterday with David Page and Brian Amani, touching on some of those caregiver and patient attitudes and perspectives towards gene therapy. I think clearly, big questions around predictability of factor level and durability, really important. I think something that's been brought up in this symposium around the ability to redose. I think that's obviously going to be a really important future consideration. I also think, interestingly, the sort of short-term use of corticosteroids as well. And I think anecdotally, hearing from peers and members in the community about their experience during that phase while on those particular drugs, I think, again, just having an expectation of what may happen during that period and whether, what type of support you may need during that phase. So I think, again, there's several big considerations, and I think it comes back to the main point that I hope, I've sort of driven through my presentation, which is about, ensuring that we have got equitable access to information and the right appropriate education for members of the community. So yeah.
Thank you very much for your very nuanced response, Lawrence. And also want to thank everybody for their participation, including the delegates that we could not see, but we could definitely appreciate your interaction online. Don't forget to fill in the evaluation form at the end of the symposium. And even though this year's WFH symposium and this satellite symposium is virtual, paradoxically, I felt very much connected to the global hemophilia community. Perhaps it's because I'm being streamed here right into my home office, but that actually means that we are respecting or applying the very motto of this symposium, which is Adapt, Innovate, and Evolve. So thank you very much once again, and thank you for your participation. Bye-bye.